doing the bulk of the presentation. She's a tenured assistant professor of pediatrics for the Perelman School of Medicine at the University of Pennsylvania. Her basic and clinical research includes interest in the development of novel therapeutics for hemophilia, and her basic science laboratory studies the molecular basis of coagulation, and she's the principal investigator of ongoing hemophilia A and B gene therapy trials. And Dr. George will be conducting the bulk of the presentation, but we're also very fortunate to have with us Jay Konduras. Jay was a person with factor IX hemophilia who has undergone experimental gene therapy. He's been free from factor use for the past 4.5 years. He now resides in Kitchener, where he and his wife own and operate a bakery. So he's an Ontarian. Was actually born in Montreal, and Jay will be providing the patient perspective in this session. So sit back and enjoy what I'm sure is going to be a very entertaining and informative session on gene therapy. And without further ado, I'll turn it over to Dr. George.
Okay, well, thank you very much for the introduction. I have to say it's an honor to be here tonight. I, through the gene therapy trials, have the opportunity to be involved a reasonable amount with the hemophilia community in Toronto, and it's really been a humbling and really great professional experience. So it has kept me grounded and really, you know, motivated to hopefully keep moving this forward for patients. So you know, it's really my pleasure to be here tonight. So, you know, as was mentioned, I hope to give you an overview of, you know, where we're currently at and some of the major issues in hemophilia gene therapy. And as a disclosure, you know, I'll be clear on when things are my opinion and when things are fact. Before I start, though, what I'd like to do is, if you don't mind, a couple polling questions, just so I can understand the group of people on the other line that I unfortunately can't see in person, but it would be nice to know a bit about who you are and a bit of your background of what you already know about hemophilia gene therapy. So the first polling question just simply asks, you know, which best describes you if you're a patient or a healthcare practitioner, or perhaps somebody that does research in hemophilia. So I'll give you a second to respond. Okay, I'll move on to the next question. I've lost control of my mouse here. There we go. The next question has to do about a bit of background of gene therapy, which is which type of vector is used to deliver the factor VIII or factor IX gene in hemophilia gene therapy? Right, just 2 more questions, so we'll keep moving. When discussing gene therapy for hemophilia B, the term Padua variant refers to what? Okay, so we'll definitely make sure we cover this tonight. And then the last question is, which of the following is a safety concern of hemophilia gene therapy trials? All right. Okay. Great, and we'll cover all of these points. So hopefully by the end of the hour, you'll have a good sense of the answers and why we chose to highlight them. So I could tell there's a range of people here tonight, which is really exciting. I actually instantly always find that I get the best questions from patients. So I'm really excited to be here. I think, we'll have questions at the end of each one of our talks, but please interrupt at any time. And if any point something, the way I'm speaking it seems to be tailored to, it's, you don't quite understand what I'm saying, don't think you're silly. Like there's a range of people here and a range of exposures already to and a knowledge base. So this is really for you to learn something. So please, again, interrupt me at any point. I'm really here to help answer some of your questions and give you a background about hemophilia gene therapy. So I'll get into my slides in a moment, but just the last point I want to make before I start are my disclosures. And I'll just briefly outline them. I am part of a monitoring committee of a gene therapy trial sponsored by AVROBio, which is, it's not in hemophilia, it's for congenital disorder of metabolism. So completely unrelated to hemophilia. I've served as a consultant for Pfizer and Bayer pharmaceutical companies. But the real, I think, conflict of interest I have is that I am the clinical PI for ongoing trials that have been sponsored by Spark Therapeutics. So it's not a financial conflict of interest. It's personally that this work means a lot to me. So that's my true conflict of interest. And I'll try my best to mitigate, however you can do, internally your own bias. So, the title of this is Hemophilia, the myths, the facts, and the future. And I think that's very appropriate. I think we've come a long way and there's still a decent amount we don't know, but it's, of course, a very optimistic time for hemophilia gene therapy. So I'll try to cover what I would consider some of the really important points by that sort of a system that Byron nicely outlined for me as a general theme of the talk. So the first myth is that gene therapy will never actually happen. And so I think for many people that have followed the development of gene therapy for hemophilia, really this work was initiated in the 1990s. And this, followed the description of the factor VIII and factor IX genes in the early 80s and significant work, basic science work sort of outlining the human genome, and sort of a natural translation of this is, could we actually use a gene as a medicine? And so what was sort of a simple idea, you have an abnormality of a certain gene, and well, let's just give you back a normal, functional copy of the gene. And so the concept was simple, but of course from concept to reality is proved to be quite a bit of effort. So, gene therapy for hemophilia has been in clinical development for probably nearing 3 decades. And I think there's reasonable evidence now that we've made really pretty significant progress. And so I would say that that's a myth. So I think that gene therapy has most certainly arrived for hemophilia, is certainly a reality. These are just a number of publications that have described successful clinical trials for hemophilia A and B. And there are many more in clinical development. So we've certainly demonstrated that this is very much a possibility. So as a brief outline of where we are and some of the success that we've had and what's currently ongoing. So first talk about hemophilia B. This table is a bit busy, but I'll just go through it briefly. So there are several ongoing trials in hemophilia B. I've outlined here the 3 that are most progressed in clinical development. So there are 2 phase 3 trials. And what that means is that phase 3 trials are the trials that are done before a drug is licensed. And so, they've done some of the early preliminary safety studies and have decided that from a safety profile, the development looks good. And so is progressing through to clinical trial, which is phase 3 studies, which ultimately, if demonstrate that this is efficacious for hemophilia patients, will be licensed in an available drug outside of a clinical trial. So there are 2 ongoing phase 3 trials and 1 ongoing early phase clinical trial designed primarily to look at the safety and as a dose finding study. Those trials, one of them is complete and has enrolled all the patients and is following patients. And so we expect to have data on this quite soon. Another is still actively recruiting. I want to draw your attention. So the transgene is the gene that we're trying to use. So in hemophilia B, of course, we want to use the factor IX gene. Universally, all vectors are using a variant of factor IX called factor IX PODWA. So that was one of the questions I asked about, and I think this is an important point, particularly in the setting of hemophilia B. I'll talk a little bit about this, but factor IX Padua is a naturally occurring mutation that was described in a family from Padua, Italy. And essentially, it's a variant of factor IX that has increased activity. So the concept here is that you get more bang for your buck. And this has universally been used now, is being universally used in hemophilia B gene therapy efforts. The vector dose, you can see that we describe the doses in a exponential manner, which is not something people commonly use in day-to-day life. I think perhaps scientists and maybe physicists, et cetera, will be familiar with this. But I think what's important is not to get lost in the numbers, but to recognize that there's quite a range in vector doses being used, and that may or may not have implications for potential toxicity profiles. At this point, we don't quite know. It certainly has implications for being able to manufacture a lot of the drug to potentially treat a large number of patients. And then the last point is that, in these trials, the level of expression has been approximately around 40% in the 2 trials that are currently in phase 3, so the furthest along in clinical development. And then the other trial is early in clinical development, and has a range of expression, and we don't, it's just earlier in development, so we don't quite know exactly where the average value is going to fall. So I'll just briefly show you some information from these first 2 trials, what's currently available. So the first is a hemophilia B trial that was sponsored originally by Spark Therapeutics and is now in phase 3 development by Pfizer. This is from a paper of ours a couple of years ago. This is looking at factor IX expression over time, which on average is around 40%. You can see a range. This publication is on the first 10 patients that were treated. They had factor IX expression that was roughly around 10 to 80%, so a range of expression. From an efficacy standpoint, this is looking at the number of bleeding events in the year before vector infusion versus the number of bleeding events after vector infusion. And you can see there was, these, what I would sort of optimistically say had a really significant impact on these men's lives. So the patients, anybody that was on prophylaxis came off prophylaxis, and far and away that nearly all the patients didn't have any bleeding events. And that has been consistently observed in follow-up of these initial patients that are now anywhere between 3 and 5 years after post-vector administration. And as I mentioned, this vector has gone on now. It is in later phases of clinical development, and we optimistically expect results within the next 1 to 2 years. And then these data are from a trial that's sponsored by the company called Unicure. These data were presented actually in December, so a little over a month ago. Similar idea here, looking at factor IX activity over time. A larger number of patients, so this is 54 patients have been followed for up to 6 months. The average factor IX activity is around 40%. And you can see here an even wider range, though, of expression. So there was a single participant who actually didn't have any expression, but the remainder of patients did have expression, and the levels are between, a couple, in the patients that had expression, a couple percent versus, 100, roughly 110%. So a range of expression, but on average around 40%. And then, as I mentioned, one of the patients did not have a response to the vector at all, which had not been observed in the prior trial. And then, the question is, what did this actually mean for the patient? So looking at this graph, we're looking at the number of bleeds in the year, in the period before vector infusion, and the cumulative number of bleeds among all these been treated versus the cumulative number of bleeds after vector infusion. And the key point is that there's a remarkable reduction in bleeds. And then if you look at the number of patients that didn't have any bleeds, the percent of patients with no bleeds, it's roughly, 3/4 of patients didn't have any bleeding events after vector infusion. And nearly all the patients, 100% of patients were on prophylaxis before they received vector, and nearly all the patients have come off prophylaxis. So really, in my opinion, quite optimistic data. And then what about hemophilia A gene therapy? This is an even busier slide, mostly because there's a lot of activity in the space of hemophilia A gene therapy. What I've listed here is the sponsors of all the trials, which is, I think, commonly how people identify what the trials are. They're all using a truncated version of the factor VIII gene, which I'll talk about briefly, but in short, the same the protein that gets expressed is a protein that's been in clinical use for about 2 decades. So the protein that's being expressed is the same as, for example, Xintha and Refacto, but is notably a truncated version of the factor VIII protein. There is a range of vector doses being used. This is a 120-fold range, not 40-fold as I have here, I apologize. There are at least 2 ongoing phase 3 trials. So again, with hemophilia A, I don't have a crystal ball, but I think that there will likely be an approved vector in hemophilia for hemophilia A gene therapy. One may expect within the next year, or perhaps even earlier. There's quite a bit of variability in the results from the hemophilia A trial, and I'll go into some of this in a little bit greater detail. But there's quite a bit of variability in the degree of expression. Some trials are seeing that the expression is stable over time. Some trials are not seeing that the expression is stable over time, and it's unclear why that's the case. Some of these patients have required steroid intervention, some have not. So there's some I don't want to call it subtleties because that's sort of not fair, but there's quite a bit of variability that's being observed among the trials. But I think one important point is that among the men that have sustained expression, they've nearly all had pretty, what appears to be, at least on a preliminary basis, good therapeutic benefit. So I'll go into the details of some of these trials, but I think the important point is there's a lot of activity in hemophilia A gene therapy, and some perhaps more outstanding questions than what's so far been observed in hemophilia B. So as I mentioned, one uniform point I think among the hemophilia A trials is that if patients had sustained expression, thus far the efficacy and sort of clinical benefit seems to be quite promising. There are 2 ongoing phase 3 studies. We expect there'll be a licensed vector for hemophilia A within the next year. And then, as I alluded to, there's several outstanding questions in hemophilia A gene therapy that we know a little bit less about, mostly because we were successful first in hemophilia B. So we have, the benefit of time, a little bit earlier success in hemophilia B than hemophilia A. And there's perhaps some subtleties to hemophilia A gene therapy that are not present for hemophilia B. Okay, myth number 2 is we've got it all figured out. I think from my ramblings in the last slide, I think you probably know that my bias here is that we don't quite have it all figured out. So we've got some figured out, but not all. So I make this analogy, I think. So I think we are, the phase of hemophilia gene therapy is we're sort of equivalent to couture clothing, really sort of intricately made for each patient. But of course, to have this widely available, we'd like it to be ready to wear. So this is this is not necessarily my impression of Canadian fashion, but I felt like it was appropriate to have somebody wearing roots. So I think we're aiming to get into this transition, right? We're aiming to move from, like fashion style, high fashion clothing to something that, anybody, all hemophilia patients can benefit from. And throughout the talk, I'll sort of outline some of the hurdles that are yet to be overcome to make that a reality. But just to highlight that there's considerable progress, but there's certainly some ways to go. So some of the, the preliminary foundation points to understand about what we're doing with hemophilia gene therapy. So the first is, I've used this term vector. What does that mean? For the purposes of hemophilia gene therapy, essentially a vector is a generic way of saying it's a way of delivering our DNA. And the type of vector we use is something called Adeno-Associated virus. So AAV, for the purposes of what we're talking about, is really just a type of vector that we use. AAV is naturally occurring in the environment. And we, what we do is we bioengineer out sort of the genes that are present in the naturally occurring AAV, and we insert the parts that we would like to express either the factor VIII or factor IX protein. So the AAV vectors are derived from a naturally occurring DNA virus, which is AAV, and AAV is a member of the parvovirus family. So if there's any virologists out there, that's sort of the nomenclature. And then I think from the standpoint of safety, AAV was initially attractive because it's not known to cause any human disease. So AAV is naturally occurring in the environment, but to our knowledge, doesn't cause any problems. There's no pathology associated with it in people. And then the other point from a safety standpoint is, for the most part, AAV does not integrate into the host DNA. And so that's really potentially something really important to consider when thinking about, if something integrates into your DNA, there's always a chance it ends up in the wrong spot and can in the worst case scenario, lead to cancer development. On the downside, if something doesn't integrate into your DNA, when the cell divides, it won't take your gene of interest, and that probably has pretty significant implications in terms of how long the therapy will last. So we'll come back to this point throughout the talk. So what does this mean? And Jay can probably inform this much better than I can, but I always find the vector infusions really exciting day. They usually end up being boring within an hour or so in, which is good. We like them to be boring, but they're usually a pretty exciting day. The vectors are delivered by a peripheral IV. What we do is we administer it over the course of an hour. We do kind of keep you for 4 to 6 hours post-vector. The protocols differ according to what most people do, but we do it on an outpatient basis. And so vector goes in over an hour. We monitor vital signs for about 6 hours after vector, and then people go home. The vector is administered into the circulation, and the way that it's, what we're targeting is your liver. We want your liver to express either the factor VIII or factor IX protein, which is the place you naturally express it. AAV has sort of a natural propensity to end up in the liver, and that's both due to the sort of tropism or the vector itself, but also because of the blood supply of your liver. Transduction is basically the term for how do you get the DNA into the cell. So these are intended to simulate AAV vector particles. So in our case, we target hepatocytes. So there's several different cell types within your liver, but hepatocytes are generally the protein-making factory of your body, of your liver. So we target hepatocytes. So the AAV vector particles will go into your hepatocytes, and then your gene, in this case, either factor VIII or factor IX, is generally speaking, so this is the cell and this is the cell nucleus, is generally speaking maintained what we call episomally, meaning outside in a circular piece of DNA, hopefully stably outside of your own DNA. So it's predominantly non-integrating. Now, an important point here is the hepatocyte is where your body naturally makes factor IX. Your body does not naturally make factor VIII in hepatocytes, but we are still targeting hepatocytes for factor VIII expression. And whether that may ultimately be responsible for some of the variability or some of the sort of curious observations we've thus far seen in hemophilia A gene therapy is not clear at this point. And then the protein, there are several parts of the DNA that we deliver from the AAV vector that are responsible for producing either the factor VIII or factor IX protein. And I'll go into what those elements are in a moment. So myth number 3 is all AAV vectors are the same. And so the truth is that's not really the case. So they are comprised of similar components, but those components can differ quite a bit. And so this will be the most technical slide I'll present. So as I mentioned, what we do is we bioengineer from a naturally occurring virus, AAV. So we kind of gut out the natural AAV DNA to insert our DNA of interest so that we can express the factor VIII or factor IX protein. So our target gene in our case is factor VIII or factor IX. A promoter is something that drives expression of your gene. And so in our case, we use promoters that are specific to hepatocyte expression. So let me just back up really quick and talk to you first about our target gene. So we're trying to treat hemophilia A or B, so we use the factor VIII or factor IX gene. As I mentioned, all hemophilia B trials are using the factor IX PODWA gene. So this is a naturally occurring mutation that was observed in a family. This protein has pretty marked increased activity. And so this was really attractive for getting enhanced expression in the context of gene therapy or enhanced factor activity after gene therapy. And so far, this has not resulted in any immunological consequences in the patients thus far studied. For factor VIII, I mentioned that we use a truncated version of factor VIII. So BDD stands for B domain deleted factor VIII, or sometimes you'll see this abbreviation, factor VIII SQ. basically, this is just a particular type of the truncated factor VIII protein. This is used nearly universally in all the hemophilia A gene therapy trials. And then, as I mentioned, this exact amino acid or this exact protein has been used for recombinant protein replacement therapy for about 2 decades. So the amino acid sequence that we're expressing after gene therapy is identical to Xintha Refacto. And I think in the Ontario area, I think Xintha is used quite a bit. There's this truncated version of the protein. The main reason we use it is that the factor VIII gene is big, and the wild type gene cannot fit into an AAV vector, so we have to use a shorter version. And then, as I mentioned, promoter is something that drives expression of a gene of interest. We're trying to express specifically in hepatocytes, so we want to use a promoter that makes genes express in hepatocytes. And then this gets a little bit into the weeds, but it's probably important, which is that you can express, so you have your DNA that drives the amino acid and then ultimately a protein. So the DNA sequence can be variable, but still have the same amino acid sequence. And there's some data to support that this variability between the vectors may have importance for eliciting an immune response that may or may not be responsible for loss of expression in a couple of gene therapy trials. So all this is to say is that even if you're expressing the same protein, there could be quite a bit of variability between the individual vectors being used. Okay, myth number 4. All safety questions have been answered. So I mentioned that gene therapy for hemophilia has been in clinical development for approaching 3 decades. Use of AAV vectors has been approximately, the first AAV vector was used for a cystic fibrosis trial in the 90s, but AAV has been used for hemophilia since the early 2000s. So there is getting to be a bulk of clinical information, what we know about use of AAV vectors, and it's being used in hemophilia and a variety of other genetic disorders. But the short is that we don't completely know the safety profile. The short-term safety is from thus far, and certainly in the context of hemophilia, has been excellent. So there are at this point hundreds of hemophilia patients that have been treated with AAV vector. And among those, 5 patients have developed sort of what I would consider relatively minor side effects: fever, muscle aches, headache, immediately after, within the first 24 hours after vector infusion, which didn't have any consequence and resolved. There's a single patient, and this has been observed across a couple different trials, so it's probably not specific to one trial, and probably is something specific. There's probably a potential side effect of the initial infusion of AAV vector. Again, at this point, looks to be minor. A single patient developed fever and headache and low blood pressure, which seems to be a little bit more, that's a little bit more significant of a complication, but again, did quite well. But this is 5 patients in what amounts to be a couple hundred patients, certainly of hemophilia patients that have received AAV vectors. So thus far, the short-term safety profile has been excellent. I will note, in trials outside of hemophilia, people have really pushed vector doses that are quite high. So you can see approximately 2-fold to 600 times higher than what we use in hemophilia. So much higher vector doses. And within the past year, there's been some pretty significant safety concerns that have come out of using these high vector doses. So 3 patients unfortunately developed liver failure 4-6 weeks after vector, and ultimately succumbed to those complications related to this. And then there were 2 patients in another trial. So this was a trial for a congenital neuromuscular disorder, and this was observed 2 patients in actually with the only licensed systemically delivered AAV vector. So sort of the same what we do with hemophilia. 2 patients developed liver dysfunction that ultimately recovered. But I think so there's a lot of information we don't know yet about this, but I think what this probably highlights to us as a lesson is that there probably are dose-limiting toxicity of AAV. So there are dose-limiting toxicities of every drug we take. You take Tylenol in excess, and that's a problem. And so I think what this says to me, and this at this point is something we don't quite know yet, is that you probably don't want to push vector doses significantly higher than what we're doing in the context of hemophilia, and that we may run into problems. But we have thus far not seen any short-term problems with AAV vectors in hemophilia in the range of vector doses we use in our clinical trials. Okay, long-term safety. So at this point, we don't have a great sense of the long-term safety. So this has been extensively studied in animal models, but of course we'd have success in AAV gene therapy before we could look at potential long-term safety concerns. So the main long-term safety concern that's been identified is the risk of what we call genotoxicity. So as I mentioned, AAV predominantly doesn't integrate into your DNA. There's a small portion that does likely integrate, that does integrate into your DNA that's been demonstrated in animal studies. And in a couple studies, what's been demonstrated in mice only is that some of these integration events were directly tied to the development of hepatocellular carcinoma. And this, again, has not been observed in people, but has been observed in mouse studies. And then people have similarly, there are a lot of the clinical development of AAV vectors for hemophilia were tested in dogs. So hemophilia A and B are known, occur in dogs. And some of those dogs have been followed for a long time, actually, up to 10 years post-vector. And they have not had any, they have not developed any tumors related to the vector, but there is evidence of integration in those animal models and what we call clonal expansion, which theoretically could be a precursor to cancer development, although that was not observed in these models. So what do we know from people? So the very first hemophilia gene therapy trial using an AAV vector that was delivered into the circulation, so into the bloodstream, those men had been followed for up to 15 years. And these are as a small number of patients. So a total of 4 patients were followed for up to 15 years. We recently published these data, and basically, the liver is the identified place that we could, if there were going to be any long-term safety problems, where we would expect to see something. And so we followed these men's liver enzymes and a protein called alpha fetoprotein, which is sometimes used as a screening test for hepatocellular carcinoma. And the good news actually is that these men they had healthy livers. And so, again, it's only 4 people. But they're unique in that they were the first, they're the longest follow-up after an AAV vector. And so thus far, that looks reassuring. The caveat I'll say is that these guys did not have sustained expression. And so we don't have direct evidence that the AAV vector was still in these men's liver. People have looked at liver biopsy specimens following patients receiving an AAV vector. This is for another genetic disease, porphyria. So you'll remember this was thought to be the disease that I think King George had. At any rate, this is a trial that went on a couple of years ago. They used vector doses analogous to what we use in hemophilia, and they looked at liver biopsy specimens, and there were, as expected, some low frequency integration events. But again, these patients have done quite well and not had any problems. And so in the community, we've started to organize around this. The clinical trials right now only require 5 years of follow-up. And so if patients were to have long-term toxicities, we might not capture this on a clinical trial. And so myself and others in the community have organized around at sort of leadership of the World Federation of Hemophilia, which is based incidentally in Montreal, has established a gene therapy register to try to follow people long-term to monitor for any of these events. And I think this is important, particularly in the hemophilia population, because as many of you know, perhaps firsthand, that the risk of hepatocellular carcinoma is enhanced in the hemophilia population, and that's due to the unfortunate consequences of the contaminated blood products in the 70s. So having a history of hepatitis C or hepatitis B virus is a risk factor for developing hepatocellular carcinoma. And so, unfortunately, a large percentage of the hemophilia population that's above 40 years of age had exposure to hepatitis C, which is, an identified risk factor of developing hepatocellular carcinoma. So all this is to say that this is a theoretical risk factor that's been identified in preclinical study. And I think the hemophilia population, for better or for worse, has a background risk of developing this type of particular liver tumor anyways. And so it will be very important to delineate when hepatocellular carcinoma occurs in patients, if it was due to the gene therapy vector or it was due to perhaps predisposing risk factors present in the hemophilia population. And so with that, so many of us have postulated that this was certainly an inevitability to occur. That a patient received an AAV vector on a gene therapy trial will develop hepatocellular carcinoma simply because of the known background risk in the hemophilia population. And that actually has occurred. So there was a single patient, this was announced in December of 2020, a patient on a hemophilia B gene therapy trial that a year post vector was found to have, actually on a routine screening ultrasound, actually was found to have a liver tumor that was hepatocellular carcinoma. And whether this is due to the AAV vector or due to this man's underlying risk factors is not clear. He did have pretty significant risk factors, including a 25-year history of hepatitis C and hepatitis B. Again, predisposing factors for this particular type of liver disease. I think we don't know. So far, I think that the inference is that this is somewhat unlikely to be related to the AAV vector. And the reason for that is that you would anticipate that this being a complication of AAV, that it would take many, many years. And so seeing it this soon post-vector makes me somewhat optimistic that this is not related to AAV, but I think this obviously is very important to understand better. Okay, myth number 6. So myth number 6 and 7, I'm going to combine. The first is AAV gene therapy is guaranteed to be a once in a lifetime treatment. That is most certainly the ultimate goal, but is not guaranteed for several reasons. And let me outline those. And I think this is an important point to think about and a way to start distinguishing some of the information coming out of these various trials. And then myth number 7 is factor activity is the main and only consideration for developing a gene therapy product. So let me just outline a couple of things. So the first is that at this point, we don't know how long this therapy will last. And we already know that some of the, unfortunately, some of the patients on these trials have lost expression. And that the best information that we have of what is going on, and this has been observed across a couple of trials, where patients have initially expressed the factor VIII or factor IX protein and then lost that expression. And to the best of our understanding, we believe this is due to something called a capsid immune response. So you'll hear this terminology. And basically the context is this. So stick with me, this is a bit of a technical picture, but you have your hepatocyte, which is your liver cell here again. You have your vector particles going in to deliver your factor VIII or factor IX gene to the nucleus. And then your body clears those vector particles out of the cell. And in doing so, kind of spits these vector particles, pieces of these vector particles back out. And it's possible that your immune system can recognize some of these pieces of the vector particles. And if it does, it recognizes this as foreign and reacts to it. It can actually clear that cell that has the vector particles. And so the problem with doing that is that, of course, theoretically, it could be a safety concern, which in the context of hemophilia gene therapy is not. But if it clears that cell that has the AAV vector particles, it's also clearing your factor VIII or factor IX gene, and then you no longer express the factor VIII or factor IX protein. And so this is why what has been incorporated in all these clinical trials is that you get very close monitoring the first 3 months after vector. So if this response occurs, it thus far has been demonstrated really in the first 3 months post-vector. So patients are followed really closely, and Jay will kind of tell you about his experience with being sort of harassed at least twice a week for labs in that first 3 months after vector, and what that was like. But we follow really closely because if we see evidence of this, depending on the trials, people will either be treated what we call reactively, they'll get immunosuppression if we see evidence of this, or they may be put on immunosuppression to try and prevent this from happening. And so that's what the rationale is for really close monitoring after vector. It's important to recognize that some, but not all AAV vectors in these various clinical trials have been responsive to immune suppression. So some people have evidence of this capsid immune response. You start them on steroids, they have a beautiful response and they maintain expression. Some people, unfortunately, with some vectors, have evidence of this response, get started on steroids, and unfortunately lose all expression. And the predictors of why this is the case, we don't have information right now to predict which patients will have this response. And we're starting to get a little bit more information of understanding how we can design the vectors to try to mitigate this. But again, we don't have all the answers just yet. Okay, so a lifelong therapy would be dependent on everybody having a response and keeping expression, certainly in that initial window after vector. And then, the question is, how long will this last? And really that's dependent on the fate of that liver cell. So if you have your liver cell here with, your factor VIII or factor IX gene, there's a couple fates of the cell that could happen. The first is that it could divide. And because your gene of interest is not integrated into the DNA, as the cell divides, at best, half of the cells are going to take your gene. It's probably much less than half, but theoretically, half of the cells could take your gene of interest. The cell could die. The lifespan of a hepatocyte is not completely clear, but it's thought to be approximately a decade. And then the other thing is something called gene silencing. Your body could recognize and turn off your factor VIII or factor IX gene that's kind of hanging out outside the nucleus. And if that were to happen, any one of these scenarios could result in decreases of expression. So what have we seen in reality? So as I mentioned, hemophilia A and B dogs were treated long before people were, and have been followed after gene therapy for up to 8 to 10 years, which is essentially the approximate lifespan of the dog, and actually demonstrated really remarkably durable and pretty stable expression for both hemophilia A and B. So that's quite promising. So what are we seeing in clinical trials? So for hemophilia B, the first successful hemophilia B trial, the data were published in 2011. So we have published data up to 3 years post-vector that the levels of factor IX expression are stable over time. And abstract presentations, people have reported that these same men have been followed, they're actually 10 years or so out, but the most recent report is 8 years post-vector and their levels have been stable. So that's also great news. And I think that's been pretty consistent across the hemophilia B trials, that if you sort of maintain expression outside of that first couple month window, that your levels of expression have been pretty, what we call durable or stable over time for the duration of follow-up. But again, we've only kind of had success at this for about a decade. So we'll need some time to really define what the therapeutic duration actually will be. For hemophilia A, the durability of expression is not clear. And this is a major, or in some cases, maybe the major question, I think, in hemophilia A gene therapy right now. So the first successful hemophilia A trial observed somewhat unexpectedly that the men unfortunately had they never really quite reached a plateau. They started having declining expression throughout the duration of follow-up, which at this point is now out to 4 years. And so that's been observed in one trial. There are hints of this same observation in another trial, but the duration of follow-up is lesser. And then there was some optimistic information that durability of expression is possible in factor VIII gene therapy. So there's one trial now that's has reported data up to 3 years after vector administration in a small number of patients, 5 patients, and their levels are like kind of what we see in factor IX, pretty stable over time. So this is a major question. So let me just show you a little bit of this data. So this is the first successful hemophilia A trial, sort of really seminal success in the field, trial sponsored by BioMarin. The blue, this is factor VIII activity over time. These blue boxes, it's called box and whisker plot, is the dose cohort here, which is being developed in phase 3 trial and expected to be the licensed vector dose. And you can see really remarkable levels of expression. So the average at a year post-vector is 64%. The average, though, at 2 years is 36%. So the subjects lost on average half of their expression. And this was initially observed in the first 7 patients, and then in information that was released last month, this has been replicated in a larger cohort of now 17 patients. And so what's exactly happening is not clear, but it's obviously really important to understand a bit better. But I think the important point is that the levels of expression have continued to decline over time, but you can still see the levels of expression are quite good, and the patients are having still, impressive efficacy. But I think obviously a very important scientific question to understand. And in contrast to this, these are data, full disclosure, that I presented this summer at a Congress looking at factor VIII activity over time. And this is a small number of patients, just 5 patients. They were followed anywhere to 2 to like 3, almost 3.5 years. And you can see their levels of expression look quite stable over time. And so the contrast here is not quite clear what's happening here. And I think it's important to acknowledge that the levels of expression here are lower than what was observed in the first successful hemophilia A trial. And so this is just a contrast that this durability of expression is a little less clear in hemophilia A gene therapy. But there appears to be proof of concept that you can certainly have durability of expression. And then, why do we care about this so much? And the reason for that is, in brief, is that to the best of our understanding right now, and this has not been proven, but to the best of our understanding, you probably, and in the current state of science, you probably cannot receive a repeat vector infusion. So not only would it be the goal that this would be, an ideally one-time or, multi-decade-long therapy that would have efficacy, it's probably obligatory right now as a one-time treatment. And that is not proven, but let me explain why I think this. So these pre-existing AAV neutralizing antibodies, so AAV is naturally occurring. And so the actual incidence of these pre-existing AAV neutralizing antibodies before vector infusion is quite variable. Because it's not standard how we do the assay. But let's just say 30, it's reported all over that, but let's just say 30 to 50% of people have pre-existing AAV neutralizing antibodies. We know from prior studies that if we give AAV vector to patients that have high levels of these neutralizing antibodies, the therapy doesn't work. So the concept would be that if you infuse a vector into the systemic circulation, this is supposed to be these pre-existing antibodies, and these are your vector particles. These antibodies would bind the vector and prevent it from getting into your liver. So currently, we actually exclude anybody that has these high levels of these neutralizing antibodies from nearly all clinical trials. Again, this is not standardized how we do this, so we don't, you can't, it's a little bit apples to oranges across the trials, but the general principle is overall reasonably supported. But what's really important is that after you administer an AAV vector, patients universally develop really high levels of these neutralizing antibodies. So in a way, we give you an AAV vector to express factor VIII or factor IX as a side effect. It's actually a very effective vaccine against AAV. And then this is really busy, but all I want to say is that you develop antibodies to the vector type that you were given, and it looks like it cross-reacts with several other AAV vector types, which we call serotypes. And the data I was showing you before, where the first patients have been followed for up to 15 years, again, a small cohort of patients, but basically not only did they develop all, and this has been shown across all trials, so everybody develops these really high titers, but these men were followed up to 15 years, and these titers, these neutralizing antibodies persisted up to 15 years. So everybody develops them, and it looks like you develop them, they're sustained for a very long time, presumably throughout your lifetime. And so just going back to the original point that durability of expression is probably very important as we currently stand in the state of science, because it looks like you probably cannot be given a repeat AAV vector administration. I will note that there's 2 ongoing trials that are enrolling patients that have these pre-existing AAV neutralizing antibodies that will, I think, help to answer this question a little bit better. So bottom line of all these points, I kind of gloom and doom here, but I'm telling you that theoretically, you could not respond, you could lose expression within the first couple of months after vector administration, or, there's some variability in the hemophilia A trials of whether or not this therapy is durable over time. I think the role of neutralizing antibodies makes the decision of which AAV vector you get very important, in my opinion. And so I think of gene therapy versus other therapeutics for hemophilia a little bit like the difference between dating and getting married. So stick with me on this Wednesday before Valentine's Day weekend. So, if you don't like your factor product, you can switch your factor product. If you don't like Hemophilia, you try something different, right? So I think that's a little bit like dating. If you get your gene therapy product and you don't like your result, you probably can't get another gene therapy product. Now, obviously there's other options if you don't like your marriage, but for the most part, it's probably pretty hard to get out of your marriage. So I think that there's a little bit more gravity in picking which AAV vector you ultimately will receive because of these role of neutralizing antibodies. So I just have a couple more slides, which is, as you recall, one of the myths was, this is a once in a lifetime therapy. That's certainly the goal, but there's several outstanding factors at this point. And then the second point was, I think there's a lot of emphasis on what's the exact factor level. And as you can see, and this is my bias, I think there are a whole compilation of other really important issues that go into, which is theoretically the ideal vector. And I just think the sort of target level of expression, you certainly ideally would like to have levels, if you could, in normal range. But if you get them in normal range, but they don't stay in normal range, or a certain, may or may not respond to the therapy, there's several different factors I think that go into this. And I think there is a defined window. I think that theoretically we can start to coalesce around, and we recognize there's quite a bit of variability in what we see in levels of expression. And so I'll just propose this as a minimal target for factor activity expression. And this is certainly my opinion. So this is data from a natural history study looking at the number of joint bleeds relative to levels of factor VIII activity in hemophilia patients that naturally have these levels. So this is looking at severe hemophilia, moderate, and mild hemophilia A patients. And if you get to factor VIII activity above 10% or so, these patients essentially don't, far and away don't have spontaneous joint bleeds. So to that, combined with the experience with emicizumab, which has been, in many ways quite remarkable. So emicizumab, the exact factor VIII equivalency is not completely clear, but it's thought to be approximately around 10%, and that's based on data at this point really in a humanized mouse model, so it's certainly not perfect. But I think basically what we know on natural history data and what we know from the experience with Hemophilia, as a minimal target level, I think that we would certainly like to keep people above 10%. And then I think the other point is that we don't want to push things too high, and that this is real busy, but basically I just want to make a couple of points. This is looking at factor VIII activity versus your relative risk of having a blood clot. If you have factor VIII, factor VIII activity above 150% is considered high. So if you get factor VIII activity levels above 150%, you start to get pretty high risks of having blood clots. And so so there is such a thing as too much of a good thing, so to speak. And I think that's an important point to remember. Actually, the data in factor IX is that the risk of blood clots at levels that are, slightly above 150% is actually less than what's been observed in factor VIII. So anyways, I think that's the general window we should shoot for. We recognize that there's a range of expression, and we probably will have to tolerate some reasonable range of expression. It certainly could be tighter than what we've so far observed. So I would argue that the ideal gene therapy would obviously it needs to be safe. You have to have efficacious factor expression in that range I've sort of outlined. The expression should be durable. It's hard to define exactly what the minimal level of acceptable durable expression should be. I think you want to have predictability that everybody will respond if they receive the AAV vector. And you want to use the lowest possible vector dose. And the reason for that is that probably if we push vector doses above what we are being used in some of the trials, we may run into short-term safety considerations. And theoretically, the long-term safety of genotoxicity appears to be correlated with vector dose. So ideally, we'd like to use the lowest possible vector dose. So in the future, these are the last couple of slides, but in the future, where are we going with this? I think from the patient perspective, I think it's been really amazing to see some of the men I've had the chance to follow. And many of them are living their life essentially free of their hemophilia, so much so that they kind of forget that they probably need to still be monitored. So you still don't forget about your hematologist. You probably should still have a factor level once a year after you get AAV gene therapy. And then you probably still need some monitoring for this theoretical safety concern to make sure your liver is healthy. And then who is the right patient for gene therapy? And, that's a discussion, and maybe this might prompt some questions, I think, when, I think maybe Jay could answer this a bit better than I can. And then the last thing I just want to mention is, access to this. So we know that gene therapy is coming with astronomical price tags. So just to give you a sense of this, these are the three of the first approved gene therapy products. One was for leukemia. Another was Luxturna is the first approved AAV vector for a form of congenital blindness. And then Zolgensma is another approved AAV vector for a fatal neuromuscular disorder. And these are their price tags in US dollars. So really impressive numbers. How do you certainly can't put a price on a disease and life altering therapy. And so, how do you start to rationalize this, and how do you prove value of this, and how do you actually bring this to patients in a way that's feasible? In the United States, hemophilia care is very expensive. On average, it's about $500,000 a year. And so there have been some cost analysis that theoretically gene therapy, if it lasts for a certain amount of time, could be cheaper than current therapy. But, is that really the appropriate way to be, looking at this? The expectation is that the gene therapy vectors for hemophilia will be quite expensive, on the order of perhaps more than $2 million US dollars. And I just want to briefly, people have started to model this and try to figure out a way of how do we actually bring this to patients, because, I can talk about the science of this, but it doesn't matter if we can't actually get it to people, right? So this is just looking at, and this isn't a problem, the good news in a way is that this isn't just a problem for hemophilia. So it really is a systems issue, and this gene therapy has become a bona fide novel therapeutic drug class that is going to really sort of test the system. And so this is looking at the number of gene therapy products relative to the year. And so we expect by the end of next year, there'll be 10 to 12 approved products in the United States. About half of them will be for B-cell malignancies. By 2030, we're expecting up to about 60 products. A lot of them, again, will be in cancer immunotherapy, but a lot of them will be AAV vectors for congenital disorders. As you all know, you don't typically make a budget over the course of 50 years. You usually make a budget for 1 year or 5 years. And so, even if something is cheaper, a one-time therapy is cheaper over the lifespan of someone's lifespan, like how do you incorporate it into that initial budget? And the gene therapies are being used for a variety of disorders. And this is a group at MIT that's breaking down this analysis. So they've talked about what this means in terms of fiscal implementation. So about half of the therapeutics, gene therapy based therapies that are quite expensive will be for oncology. Half of them will be for ultra orphan disorders. So these are disorders, much rarer even than hemophilia. So those are a small number of them. And then these so-called budget busters are perhaps where hemophilia may fall into things, which is, there's roughly 20,000 in the States. I know the States numbers better than Canada, but there's roughly 20,000 hemophilia patients in the United States. Let's say half of them are severe hemophilia patients. So how do you bring this therapy for, thousands of patients at greater than $2 million? And that remains an outstanding question. I will say this, I think there's a couple good news about this in terms of how to actually make this financially possible. In Canada, I think this is potentially, from an implementation standpoint, may work a bit better than here in the United States. So these issues, I think, are probably perhaps better tackled by a system as a universal healthcare system. Number one, a universal healthcare system is much better poised for negotiations with pharmaceutical companies on pricing. The second is, if a therapy is going to be $2 million and 10% of people don't respond or they lose expression quickly afterwards, like you would ideally not like to pay for that. In the United States, that would be hard because there's there's so many different insurance companies that people switch insurance companies quite readily. But in a universal healthcare system, it seems reasonable that you could more easily enact a system where if therapy doesn't work, you don't have to pay for it. This has been actually used for the drug Chimera, which is used for B-cell malignancies. And this is the policy that if the patients don't respond, then the therapy is not paid for. And then other people, the ideal situation would be annuity payment so that it's not this huge influx of money that's required. The thought about that is that this is going to be very hard in the United States. So on average, in the US, hemophilia patients switch insurances every 3 years. And so keeping track of people might be a formidable difficulty. And obviously, there's not a motivation on the behalf of the insurance company or patient to be paying a bill. And so, but at a universal healthcare system, this theoretically is something that could be enacted and maybe quite effective at making this available to a large number of patients at the same time. So, as I mentioned, because hemophilia care is so expensive, theoretically gene therapy could be cheaper. And the question is, how do you assign value? And the truth is, I don't really know. I think it's reasonable assumption that it doesn't cost $2 million to manufacture the vector or to distribute the R&D costs into that. But what the cost should be is quite honestly not something it's beyond my level of expertise. But I will say, in the United States or in many developed countries, the way a drug is priced is the value relative to available therapies. And so in a way, that's kind of good for hemophilia because available therapies in hemophilia are so expensive. And this was an interesting problem, so to speak, when Luxturna was developed. So this is an AAV vector for congenital blindness, which costs about $800,000 US. When this was, when this was brought to insurance companies in the United States, it was either they pay $800,000 for these patients so that they could maintain their vision, or they could pay nothing because there's no other available therapy. And so that was a quite complicated, negotiation. And in a way, because hemophilia care is already so expensive, there's already a precedent set and a budget aligned for hemophilia patients. So I'll stop with this, just to outline that I'm obviously quite biased, but hemophilia gene therapy has demonstrated really significant promise. And we have several outstanding questions. We have to tackle this issue of AAV neutralizing antibodies so we can treat more people. And so we theoretically could try to re-administer vector. I think this will be particularly important for pediatric patients, I'm a pediatrician, so this is near and dear to me. We have to better understand this capsid immune response and universally be able to have people be able to maintain expression. We have to better outline what the durability of expression is and what our expectations for it is. And I think the other point is, better understanding the predictability of response among patients. From the standpoint of a patient, I think it's very important to choose wisely at this point of what the right vector is. But, as I've outlined here, there's considerable progress and considerable efforts being made for gene therapy. So I think some of these issues very much will be tackled. How long it will be is hard to speculate, but these are important issues for the success of the therapy. And then the last thing I think is important to keep in mind is that people are using AAV for many other disorders. And I think sometimes in hemophilia or in everything, really, we can get quite siloed, but we can actually learn a lot about what's happening and how these therapies are being implemented for a variety of other genetic disorders. So I personally am trying to reach out to people that are involved in AAV gene therapy for other disorders so that we can kind of work together and try to, many of these issues are not specific only to hemophilia. And so I think that's also a sign of optimism because we can draw not just from our own community, but we can draw from the community of several congenital disorders. So thank you very much for listening to me. And with that, I'll turn things over to Jay. I just have to say, I had the opportunity to, I follow Jay and his brother, who are both enrolled in our trial. They very nicely agreed to take part in a New York Times article. And it's always just so humbling to hear the patient experience. And so I think this was your brother, Jay, that described the therapy this way. But, it's really been an honor to be part of your care, Jay, and part of the community. And I think Jay's experience will speak more to what this is really all about, certainly, than what I've shown you on these slides. So I'll turn things over to you.
Okay, thank you very much. I'll just spend a few minutes so everyone can do questions, but I'll just explain who I am and really briefly why I chose to go on a trial, and then quickly about the trial itself, and then what it's been like since the trial. So just to get you a frame of reference of my, where I am on the hemophilia kind of like injury spectrum. So typically I would get 4 to 8 injuries a year. I'm assuming that a lot of people on this call know all about injuries, so I won't detail it, but you know what it's like. So about 4 to 8 a year, that's where I'm at. That's historically through my life. And then I've been, like, I'm 56 years old, so I've been through all of the plasma factor 9 and the recombine it in the 1980s and all that stuff. So I've got through all that. And then I'll just mention, like getting right to why I would do a trial, even though the recombinant got to be very, like a really good treatment, and you didn't have to worry about pathogens and stuff anymore. In 2014, I actually hurt myself just shoveling snow. So something kind of what you're always doing in life, and ended up really injuring myself badly in my leg, and ending up 10 days in the hospital, then a whole bunch of weeks of recovery, and then into months with the canes and crutches. And then it was really like 6 or 7 months, before I'm back kind of normal again, even though you have to then build up your leg muscles again. So then right after that, that puts you into a kind of like depression mode that you just can't, you can never be sure that you can control it. It's always there hanging over your head. Anyway, so in 2015, I was able to get the Harvoni because I had the hepatitis C from some, whenever in the past. So then that kind of was super, that's super successful. And then a few months after that, I got a letter from Dr. Title in Toronto, that's my doctor, just offering opportunity to go into a gene therapy trial. So you can picture my view as like, sign me up, where do I go? Because my view is like, let's do it. And also because someone's asking you to do a trial, so it's like, okay, well, then they must need help, right? So that's all, that was kind of into the why. It wasn't much more than that to make me decide, because you have a whole lifetime of reasons why. But then after that, I did do an awful lot of research. There was less going on at the time, but still quite a bit related to gene therapy and AAV, and I was somewhat familiar with it, even though I never really followed cures. I just was living life, figuring out, just go along with what was available. But I did a lot of homework on who's doing the trial and just what the trial was all about and what it was involved. So I was like super impressed by everybody and everything that I could find about the trial, and talking to Dr. Title, and then even Dr. Catherine High, who seems to be a huge lifetime history in working the hemophilia, and then meeting Dr. George and having all my questions explained. So it was all still green lights all the way for the trial. And then just on the trial, it was just, I suppose it's a bit nerve-wracking, but I was very confident and at peace with what was going on. So I wasn't scared at all. So the trial, the treatment itself is like literally it's 1 hour with an intravenous drip. It's truly a non-event. And yeah, a boring day was a good day. So it was that my wife and I watched movies for the afternoon. So it was that non-eventful. And then so it's pretty intense with blood draws every couple days. I can't remember, because it was just fun, because every time you got a blood draw, you got a feedback of what your factor level was doing. And it just, in my case, and a lot of people's cases, it just creeps up. And if your factor level is creeping up on its own like that, like who cares about blood draws and stuff? It's like, it's all part of the fun. So the trial itself for me was intense for about a year, and then it's been follow-up, and I'm still on the trial, but in the follow-up phase, which is a lot easier, like a few blood draws a year. So the trial itself has been a non-event, other than it's been like the best time of my life, and we would get to fly down to Philadelphia, and the people are super friendly and nice there. So the trial itself was, again, it was like a really enjoyable time. And then since then, I don't know how to I'll explain it very briefly, but I haven't had any bleeds in like 4.5 years. So you can really literally say no more, like you're a kind of a normal person. And I've had lots of instances of damage in that period of time. I don't write them down or anything, because you'll hit yourself, and you know that, okay, that would cause a bleed. And then just every time it's like, you're better within a few hours. And then the next day, when you'd usually expect something to be like 5 times its normal size or whatever, it's like, nope, you don't even feel anything. So every day to me, like, and it's, I'm appreciative that I have the gene therapy. And if I were to say anything about side effects, I would say the only side effects I've noticed is that in the mornings I wake up and I don't feel stiff, or when I'm getting up out of an airplane to walk somewhere, or when we could do that, or a movie theater, after a few hours of sitting there, I don't, I have a bad ankle historically, and I don't feel stiff. So to me, that's like, I wasn't expecting that kind of like benefit, but that's really nice side effect. And yeah, the other thing too is noticeable is I've had way more endurance in standing, walking around for like 6-8 hours a day without sitting down, where before I would always have to find a chair after a few hours and sit down for 15-20 minutes and then get up and go again. So I find my physical endurance has been noticeably better. So if you consider that a side effect, again, those are things I wasn't expecting that would be the result of this, but so that's about it. That's it. So the punchline is I haven't had any treatments in 4.5 years. So that's my story. Thank you.
Thanks so much, Jay. We really appreciate it. So now we'll get to the question piece. I've got a few that have come up during your presentation, Lindsey. So I'll deal with one here, and then you've got a bunch that were already sent in as well. So I'll just take this one off, and then we can go to your list. It's, hi, does the gene therapy affect the next generations in the family? My son has severe hemophilia A, and I'm wondering if the gene therapy will change the chance of his kids inheriting hemophilia.
Yeah, it's a really good question. So the short is that he would still pass on the affected gene, meaning his future generations would be at risk of still having hemophilia. So it does not change that. And the reason for that is that the gene is outside his DNA, and so it doesn't change, the inheritance of the gene. And that's a really good point, though.
And let me just go over to the chat. It was a real short question when you're on the point of some of the conditions that need to be met for gene therapy, and you were talking about Hep C, and the short question was, what if somebody had cleared Hep C?
Yeah, so it's a great point. So if you, so right now on the clinical trials, patients that have active hepatitis C are not eligible. But if you don't have active hepatitis C, meaning you've had it and you cleared it, people are eligible for the trial. What it will mean if you have active hepatitis C and you once the drug is licensed and you can go to your doctor and get it, is not clear. And then I guess the second point I made, I kind of spent a decent amount of time talking about the background risk of developing liver cancer in hemophilia patients because of the risk, so many patients unfortunately contracted hepatitis C. And, as an aside from gene therapy, because of the sort of remarkable success curing hepatitis C, we don't know what the long, we have data from, 20 years ago, what it meant to have hepatitis C for 20 years, but we don't know what it's like now if people had hepatitis C for 25 years and took Harvoni or some of the other drugs that came out, 5-10 years ago to clear hepatitis C, and what that future risk is for developing hepatocellular carcinoma. So that's an important question. So I guess it's tiered. Right now, we're not enrolling hepatitis C positive patients. We enroll people, we enroll lots of men that have had a history of it, but then cleared. My guess is that with more comfort with the therapy, that patients perhaps that are hep C positive may ultimately be treated. But I think we need to understand a lot more about, these risk factors that have been identified of this potential risk of developing cancer that may not be a real risk in people.
Another question we had, Lindsey, with respect to clinical trials, have they all been with adult males? Any with children done to date on the horizon? And there was another question directly related to this one was, what would be the youngest age to be considered for a gene therapy trial?
So it's a really great point. So right now it's only adults, so 18 and above. There is considerable discussion to extend this down to teenagers, 12, 14-year-olds and above. Treating, ideally, this is a disorder you're born with. Ideally, you treat a baby, right? The reason, so there's two reasons about why babies right now aren't considered. The first is, I think everybody wants to have a greater level of comfort and understanding and, better knowledge base, certainly before they treat a baby. But we know that because the gene doesn't integrate into your DNA, as the cell divides, you'll basically effectively get lower, as your liver is growing, you would get lower and lower levels of factor VIII or factor IX activity. And so if we treated a baby, we know that they, over their lifetime would have lower and lower levels of expression. So the current methodology, we probably cannot treat children unless we've developed the ability to either re-administer the vector safely or we kind of come at this with a different strategy.
Great. So those are the questions I've got up on Q&A and chat. I don't know if we can move to the pre-questions that went in.
Sure, I don't.
Kaylee, maybe you can help us with.
Yeah, no problem. I did see a few more come up as well on the chat. I'll pull up those other questions, so no problem.
I think I can see them. And I apologize, I started answering them because I realized I forgot to answer them after we spoke, but is it okay if I just try to answer these that are in the chat really quick? Is that all right with you guys? Mm-hmm. Okay. So someone asked a question and this, I was a little afraid that sometimes I would use too technical of language. So expression just simply means we do our gene therapy and we can detect factor VIII or factor IX activity when we do a blood sample from you. So expression basically just means the therapies, you're actually making the protein from the gene therapy that we gave you. So that's a really good point. And I appreciate someone clarifying that. And then I think somebody asked, so somebody asked about kind of on the point of that right now we're not treating pediatric patients. Somebody else asked, if I have mild hemophilia, are we treating mild hemophilia patients? And right now the answer is no. And this is actually a topic of hot debate, because it's like if you treat a severe or a moderate patient and then they have a factor level of 40% or 100% or 10%, and somebody has mild hemophilia and their baseline levels 5% or whatever, it seems reasonable that we should extend this out to anybody that has either wants this or certainly people that have bleeding in the mild range. And I think I think the short answer is that there's pretty significant advocacy for that. It just hasn't happened yet. And I think right now, because we're all in clinical trial, and so the target population is to treat the severe or moderate patients. But I I'm optimistic it will extend up to mild patients. And I think that's a really good point. And then I think somebody asked a question that makes a lot of sense. Someone asked, hemophilia A seems to be a work in progress. If you had to choose to get a therapy, would you proceed at this time? And maybe, I don't know, I might toss this over to Jay to see what his thoughts are. Put you in the hot seat. Oh, I think you're on mute, Jay.
I didn't understand the question.
The question was, if you had to choose right now, would you go forward with gene therapy, and kind of what would be your mindset in picking which trial to?
Oh, yeah. Okay. Yeah. I guess it gets back to what you said. When I was deciding, like, it was emotionally, it was like, I'm all in, but scientifically, I better read up on this to see what I'm getting into. So at the time, there was a lot of information, but there was only a trial or two, coming up to speed. Now, like 4 years later, there's a lot more information. So like you had said, I can see myself being maybe more picky now that I can see. Personally, my kind of, the thing I see is the most problematic is the dosing of AAV, which I think I wrote you a letter a long time ago saying, thank you for being such a conservative trial, because some of the other trials were like really highly dosing people. So, but now you can see the kind of like effects of that. So yeah, I would be more picky, but I would, there's so much information that you need to learn a bit about the science, but again, there's a lot of resources for that too, but I would just do more homework. And then the answer would be yes. What I've seen out there now is I would still pick one.
Yeah, I agree with the general sentiments, Jay. I mean, I think the short answer to that is to choose wisely and to make sure you've got all the information at hand. And I think when we went through this, I think we spoke on the phone multiple times. It's just important to not have this be a rush decision, that this is, you have all the information in front of you, and you know what we know and what we don't know. And it's like anything with that, there's no guarantees in life, but to have all the information available to you. Yeah, you guys have great questions. So somebody asked this, so for hemophilia A and B, we both target hepatocytes or the liver cell, and why do we do that for factor VIII gene therapy if we know factor VIII is not made in hepatocytes? The short answer, I think, is that it's easier and we can do that. But I'll just extrapolate that a little bit. So we know hepatocytes are long-lived cells. We think that the cells that factor VIII is naturally made in are not as long-lived. And so theoretically, the durability should be longer in hepatocyte expression. The other is that when you design the vectors, the part of the vector that actually drives you to express the protein or make the protein from gene therapy, called the promoter, we've kind of bioengineered the ability to express things from those promoters specifically in liver cells. And so it would take, there's a lot of work ongoing that's in preclinical phase that maybe you could try to express from different cell types. But the short is that we're the best at expressing things from the hepatocytes or liver cells.
Lindsay, I've got one. Gene therapy, and he's got GP1B, possible in Bernard-Solier syndrome affected and/or carriers.
Yes, so that there is some preclinical work ongoing for that. People are using a totally different approach than what we do for hemophilia. And, because you want to target the platelets, which platelets live like about 10 days. So AAV is probably not the way to go to do that. But people are targeting that. So it's a way of targeting the stem cells in your bone marrow to properly express the protein. But yeah, there is ongoing work on that. To my knowledge, not in clinical trial, but some preliminary success in early phase, early preclinical studies.
Another one, are you concerned that the drop in factor VIII expression from infusion to year 4 in the BioMarion will continue?
Oh, geez, I don't have a crystal ball. I mean, I think, I wish I did, right? Could have been the lottery, but I think that what we know so far is that the levels continue to decline each year. And so, you know, I mean, I guess the best predictor of the future is the past. And so, but the only caveat I'll say, and this gets a little in the weeds, but in short, there's some thought that maybe the factor VIII protein itself is kind of like making the liver cells not that happy. And so perhaps if you get down to lower levels of expression, then the cells might be happier and it might stabilize. But that's completely unfounded. So my best guess is that it would probably continue to decline over time, but I think we just need more information and more time to know.
I have another one. If a patient was on this therapy, how would the doctors medicate if a patient required surgery, example, joint replacement?
That's a good point. So this actually come out quite a bit for, it seems to me that people come on the trial, then they are more active and then they want like their knee replacement and they sort of fixing themselves up a little bit, if that makes sense. So there's been a couple ways. So I think the first thing that we know that we, you know, again, these are small numbers, but with respect to using the potable protein, which is again, sort of a gain of function protein, it looks like it, you know, when we measure the factor IX activity, the patient's bleeding pattern behaves like the measured factor IX activity. So I think that's reasonably predictable. So we've had patients who have levels around, you know, the 30 to 40% that got knee replacements that their factor level was corrected, you know, back up to 100% at the time of surgery. We've also had patients have pretty significant surgeries without any factor replacement. And so I think it's not standardized by any means. My conservative opinion, if I were to pick, and if you have control over the situation, is that if someone has you know, if you're still in the mild range of hemophilia, that we would treat you like we would treat a mild patient. And if you're going to have a knee replacement, you would get some factor.
Great. So given what I'm seeing left, you've covered, I think you've answered all the questions through your presentations and then outright just now. So I'm not seeing anything else unless Kaylee, you have something I haven't seen.
That's all I have for the moment, but encourage any attendees to submit any additional ones. Oh, there, you've got a plug for your bakery, Jay.
Bakery's fantastic. He and his wife bring us great treats when we
So the question, Jay, is are you willing to share the name and location?
Yeah, sure. It's not related, but it's the Nougat Bakery, Nougat Bakery, and we have 2 stores, one in Waterloo, one in Kitchener, and a bakery in Kitchener. So
Great.
And that's where I do a lot of hurting myself lifting flour bags and things.
Okay, well, that looks like we've covered it all off. So I just want to close by obviously saying thank you so much, Dr. George and Jay, for giving us your time. It was a very informative presentation. We are going to have it vetted and up on our YouTube channel probably within the week, and we will send a blast out to our membership and put it on the website. So for those that might have heard about this and didn't have a chance to participate, they'll at least be able to go and check it out, the entire presentations, probably in a week or so. So thanks so much to everyone who attended. Hopefully you got all your questions answered. And again, thanks to our presenters for a really informative and down-to-earth presentation.
Thank you for the invitation.
And Kaylee, unless there's anything else, I think we will sign off.
Great. Thank you all again, and enjoy the rest of your evening.
Thanks so much.
Bye.
Thanks, everybody.